Lymphocytes. Generally, lymphocytes are smaller than phagocytes. But even if they are way smaller than phagocytes, they have very big or large nucleus. They are specialized to respond to one antigen alone. So you could say that they are specialized. There are two types of lymphocytes. We have B lymphocytes and T lymphocytes. B lymphocytes are also known as B cells. B lymphocytes stays in the bone marrow until maturity. They could be seen in the lymph nodes and spleen, but there are some found in the blood. Their primary function is to produce antibodies. Memory B cells store the information on how many specific antibodies needed to tackle specific antigens. They then divide into plasma cells which have thousands of antibodies that combine to the antigen. So now let us see how B lymphocytes work. We have a pathogen and we have an antigen presenting cell. Once a pathogen becomes present, an antigen presenting cell displays antigen with the help of proteins on their surfaces, and this process is known as antigen presentation. This process had to be done as it allows for specificity of adaptive immunity in lymphocytes. We have here the antigen again on the cell surface membrane of the antigen presenting cell. Antigen presenting cell could either be a macrophage, a dendritic cell, or another type of B cell. In this area, however, we have a B cell that is not activated, and we call those inactive B cells as naive B cells. Call this one naive because these B cells do not know anything yet, meaning they have not met or encountered any antigen in their entire existence. The interaction between the cells that we have in here leads to the activation of the B lymphocyte. As the B cell is now activated, it now undergoes differentiation and can now produce the plasma cell and memory cells, as you could see in the arrow going down. Plasma cells are important as they have the ability to create antibodies. Memory cells, on the other hand, are produced over and over and over again, and it can survive for decades. They only carry specific receptors throughout life. The receptors will only recognize the antigen that they have encountered when they became activated. They are important in cases when there is a case of reinfection. This means that when the memory B cells re-encounter their specific antigen, they proliferate and differentiate into plasma cells, which then respond to and clear the antigen from the pathogen. This concept is important in understanding how vaccination and secondary response works later in this chapter. So let's put this one into another perspective. Let's say the bacterium in here is a type of virus, maybe. Yeah, let's say it's a virus that you haven't had before. And let's say it's a new strain of cold virus, maybe. Okay, so since this new cold virus is new to your body, there will be no recorded information to when this new cold virus goes into your body, a macrophage, a dendritic cell, or another type of B cell will be processing it like this, and it will make sure that the antigens are displayed outside the cell. Okay, then once these antigens are outside the cell, by random chance maybe, there will be a naive B cell, meaning again, these are B cells that have not had encountered any type of antigen before. So this naive B cell becomes activated. And when we say activated, it already has its own data, and this will recognize this new strain of cold virus over and over and over again every time it goes inside or your body. So once this B cell is now activated, it will be creating plasma cell that produces antibodies, and then later on it will be creating, or it will be multiplying as memory cells that will remain in the blood. It will be there in your blood. It will be surveying the area if there is a presence of this new strain of cold virus in the future. Question. B lymphocytes have large number of mitochondria, extensive rough endoplasmic reticulum, and several Golgi bodies. Explain why this is so. The reason why B lymphocytes have so many mitochondria, so many rough ER, and several Golgi bodies is because they need energy for protein synthesis and secretion. They need the endoplasmic reticulum for synthesis of proteins, and the Golgi bodies will be preparing those proteins that were created in the form of antibodies for secretion.